How do proteins in your cells transmit signals to one another? There are many types of signals and many ways of transmitting them, and this is the first part in a series of videos where we'll explore some of the ways that cellular signaling works. Today we're going to look at phosphorylation. When we say phosphorylation, we're talking about the transfer of a phosphate onto a particular amino acid within a protein. So let's start at the beginning and have a look at what a phosphate is and where we can get one. Your cell's source of phosphates is a molecule called adenosine triphosphate, or ATP. ATP is a source of chemical energy, and the production of ATP is one of the things that happens when we eat food and breathe oxygen. ATP consists of an adenosine part and 3 phosphates, called the alpha, beta and gamma phosphates. While the chemical bonds in the adenosine part are really strong, the phosphates of ATP are much less firmly attached, and some proteins are able to pinch off the gamma phosphate and stick it on the end of a particular amino acid. This is called phosphorylation, and it's a way in which proteins transmit chemical signals to one another. Out of the 20 different amino acids in your body, there are only 3 that can get phosphorylated: serine, threonine and tyrosine. Technically it's 4 because histidine can get phosphorylated as well, but it's more of a bacterial thing, it doesn't happen that often in mammals, and the mechanisms of it are frankly a bit confusing. The proteins that are able to pinch a phosphate off of ATP and stick it onto a serine, threonine or tyrosine are called kinases. There are around 518 different kinases in a human cell, so you'd expect them to be split into 3 groups for tyrosine, serine and threonine phosphorylations. But that's not exactly how it works. It turns out that serine and threonine are pretty much indistinguishable for kinases, so the ones that can phosphorylate a serine can also hit a threonine. Apart from serine-threonine kinases, you have the tyrosine kinases, and there is an overlap, which are called dual specificity kinases, even though triple specificity would have made more sense because they recognize all 3 phosphorylatable amino acids, but that's scientific nomenclature for you. There's also a group of close to 50 kinases that have lost their phosphorylation activity. We call those pseudo kinases, although some pseudo kinases have a bit of activity left in certain circumstances. When a protein gets phosphorylated, the phosphate on a serine, threonine or tyrosine residue acts as a little flag to other proteins. They're attracted to the phosphorylated protein where they weren't before, and they bind on top of the phosphate. So these phosphorylations are able to cause interactions between proteins, and that way signals can be passed on all the way across a cell. Ultimately, this can lead to a variety of things to happen with the cell, like cell division, cell migration, and even cell death. At the same time, faulty hyperactivated kinases can transmit too many phosphorylation signals, and this can lead to diseases such as cancer. So let's take a closer look at how a kinase can pinch off a phosphate and transfer it across. Here you see a structure of a kinase. In the middle, it has a big pocket that ATP can sit inside. Even though there's a fair bit of variation in the amino acid sequence between human kinases, a few amino acids are exactly the same in all of them. Let's have a look where they sit and what they do. The important bits of a kinase can be split up into 3 groups: the bits that bind ATP, the bits that bind the target substrate, and the bits that transfer the phosphate across. In order for ATP to stay inside the ATP binding pocket of a kinase, it needs a seatbelt to keep it in place, and there are a few amino acids responsible for that. Firstly, you've got the glycine-rich loop, which extends over the top of ATP. Then there is a very important lysine residue that binds the alpha and beta phosphates of ATP, keeping it in place. If you mutate these amino acids, the kinase won't be able to bind ATP anymore and it loses its activity. A conserved glutamate forms a salt bridge with that lysine, and that's important for keeping the shape of the kinase stable. On the other side of the pocket, there's a DFG motif, which binds metal ions that you need to transfer the phosphate. The DFG is also the start of a large loop called the activation loop. The amino acid sequence in the activation loop determines whether the kinase recognizes tyrosines or serines and threonines. Lastly, there's the YRD motif, or HRD in many kinases. This last bit is maybe the most important part, because you need one particular amino acid, the aspartate, in this motif to actually transfer the gamma phosphate from ATP onto a substrate serine, threonine or tyrosine. So if you lose that particular aspartate, the kinase can still bind ATP, but it won't actually transfer the phosphate. So kinases are extremely important for the way cells transmit signals and regulate their behavior. Next time we'll go into a little bit more detail about some of the roles that kinases can have in diseases such as cancer, and how we can target them with very specific drugs. Thanks for watching, drop any questions you might have down in the comments below, and don't forget to hit the subscribe button. See you next time.